have a short video. So Dr. Huang from McGill couldn't join us this evening, but he put together about a 5-minute video to talk about the life cycle of RAI1. And his approach, as Gianluca was stating, is a different approach than Sarah. He's looking more at a small molecule approach, looking to try to decrease the degradation of RAI1 so that it hangs around in the cell longer. And so he'll talk a little bit about that next. And we use subtitles because the sound is not perfect.
My name is Huang. I'm a professor at McGill University in Canada. I did my PhD at Baylor College of Medicine in Texas with Dr. Huda Zobi. She is a pioneer in child neurodevelopmental disorders. Then I moved to California and did my postdoc with Dr. Li-Chun Lo at Stanford University. At Stanford, I became interested in Smith-Magenis syndrome and the molecular as well as neurobiology of RAI1. At Stanford, I discovered that if you increase RAI1 level back to normal, you are able to rescue some disease feature in a mouse model of Smith-Magenis syndrome. We also did different deletion to find out which brain regions depends on RAI1 for normal function. So building on these exciting findings, in my own lab at McGill, we are interested in studying the molecular function of RAI1 and hoping to find a cure for SMS. Today, I want to illustrate some of the ideas that we have for finding the cure for SMS. So first of all, I want to talk about the life cycle of RAI1. RAI1 was induced during development by a retinoic acid molecule. So it binds to the receptor, which translocates into the nucleus of the cell here to activate RAI1 gene expression. After RAI1 is produced, it will be released into the cytoplasm here, and it will be modified by different chemical molecules. Some chemical molecules will make RAI1 more stable, some will make them RAI1 less stable, and some will basically target RAI1 for degradation. And RAI1 will come back to the nucleus and binds to the DNA and regulate gene expression. And we know the function of these RAI1 downstream target gene is very important because if you have less RAI1, the expression of some of the genes will be suboptimal. And this is why we think it will cause disease. And knowing this, we decided to pursue 2 different strategies to increase RAI1 level or target downstream RAI1 downstream molecules. The first strategy was by identifying the different enzymes that modify RAI1 stability. We are focusing on different kinases and phosphatases because those molecules are commonly targeted by drugs. We hope to identify those molecules and use drug to inhibit those molecules to increase RAI1 stability. Our recent finding funded by SMS Foundation showed that RAI1 is highly unstable in the cells. So by stabilizing RAI1 in the cells, we hope to increase the level of RAI1 and hope we can improve the symptoms of SMS. Another strategy was to identify important RAI1 downstream targets. We know there are many different RAI1 downstream targets, so we are trying to dissect each one of them to see which one is the most important one. And hopefully we can focus on the more druggable molecules and target them for therapy in different animal models of SMS. I want to emphasize that all of these discoveries and ongoing works they were supported by SMS Research Foundation. Without the help of the foundation, it is not possible. I would also like to thank all the family for support SMS Research Foundation. Although research is difficult and challenging sometimes, I promise we will not stop finding the cure until we find one. Thank you.